Now, Sarah, you're also involved in nanotech research, but you have a slightly different angle. You're looking at biomedical products, things to use in the field of medicine. What kind of biomedical products are you working on?
Yeah, my group is really interested in trying to design new materials that can mimic the materials that naturally occur in our bodies. So throughout evolution, our bodies have evolved to have these really exquisite materials with these interesting properties down at the nanoscale. We want to try and understand how that works so we can recreate our own proteins that begin to mimic some of those functions.
What would be some of the practical applications? What sort of health issues would you address first?
So right now we have projects looking at peripheral arterial disease. So this is very common with diabetic patients. They have compromised blood supply to the lower limbs. So we're interested in creating new materials that would help them to regenerate new blood vessels in those tissues. And that's a collaboration with Professor Nan Huang at the Veterans Affairs Hospital in Palo Alto.
So you would inject cells or proteins into the body which the body would think was part of itself, and they would start functioning as if they were part of the body?
Yes, exactly. And the trick is to design the materials so that they can recreate some of those functions that healthy materials in our tissue would normally have. So cells are the smallest living units of our body, but in order to do their jobs properly, they have to be connected by a material that organizes them into a proper structure and gives them the right directions about what to do and how to function. So we want to make our materials that we can use to replace that tissue when it becomes diseased or damaged, and so it's no longer telling the cells properly what to do.
Okay. So you're creating new materials to put into the cell, and I understand you're also creating materials to help you get the new materials into the cell, because you can't just inject it, it'll all break down. How you carry it into there is very important, and I think you have some slides to demonstrate that.
Yes, I do.
So can we see the first of Sarah's slides, please? Okay, so what is this about?
Yeah, so in the top left panel, what I'm showing is a vial of liquid and a vial of gel. And that gel is actually one of our protein engineered gels that we've designed to help when you're doing cell transplantation. So cells, and in particular stem cells, are being looked at for a variety of different medical applications. But when you inject stem cells through a syringe needle into a patient, many of them die. And that's what the lower left panel is showing. We've stained these cells with either a red dye or a green dye. And when they die, they turn red. And when they're living, they turn green. If you just inject them inside of a liquid carrier, which is what most clinical trials use today, about half of them, almost half of them, die. In contrast, if you deliver those cells within a gel carrier, the gel acts like a cushion to help provide some mechanical protection to the cells as they go through the syringe needle and enter the patient.
And the gel is also an organic substance, isn't it?
Exactly. So this gel is made out of our engineered proteins. So it would be a transient, short-lived material inside the body. It would slowly degrade back into its amino acid building blocks and just be resorbed and recycled by the body naturally.
And I think we have a few more slides on this concept. Can we see? Okay, there's the next slide.
Yeah, so when we try to design these new materials from scratch, we begin by thinking what type of tissue do we ultimately want to regenerate, whether it be blood vessels, muscle, or nerve, for example, and what would be the properties that an ideal material would have? What type of mechanical or structural properties would you want it to have? How quickly do you want it to degrade after you deliver it to the body? And so we design different nanoscale building blocks that we then use kind of like Legos. We mix and match them together in new combinations to create a new functional material that we could then deliver to different tissues in the body to try and help spur on tissue regeneration.
Now, working in such a small scale, and do you have special tools? Because how could you even see what you're doing?
So you can see it at the nanoscale, and an individual protein you also can't see by eye. And so we use a lot of biochemical and biomolecular approaches in my laboratory that might be more common in a chemistry lab, actually, than a material science lab to try and understand what's happening.
Okay, and we have another slide which illustrates more about this. Can we see the next one?
Yeah, so to actually make these materials, proteins in our body are encoded within a piece of DNA, and so we use that same strategy to make our engineered proteins. We encode that information, the instructions for making a new protein in a piece of DNA, and we give that DNA to a bacterial host, and then that bacterium becomes our material making factory and makes a whole bunch of the material for us.
So you start with a little bit of protein that you take from a person, inject it into this bacteria which replicates it for you, and then you have a whole bunch.
And so what we are injecting into the bacteria is a piece of DNA. And it doesn't have to be DNA from a person. It's a completely new synthetic piece of DNA that we chemically synthesized ourselves. But the bacteria is able to make a whole lot of it. And it's a bacterial culture of fermentation, much as the way you would ferment beer, for example. It's a very similar process.
Now, is that to create the gel that carries the cells?
Exactly. That's to create that protein material that we then can fabricate into a variety of different structures. We can make them into hollow conduits if we're trying to make synthetic blood vessels. We can fabricate them into gels that would be injectable. We use them as coatings for bone screws. That was one of the images that was on the slide.
And I think you've got one more slide. Let's see this last one.
Yeah. So this is an example of how you can kind of put all the different pieces together. So if the little yellow star there is a neuron.
At the bottom of each circle.
At the bottom of each circle, exactly. There's a yellow star there. That's meant to be, to demonstrate a neuron. And neurons, as they grow and regenerate, they send out these long projections that secrete enzymes. And we want those enzymes to degrade our material so that the neuron is able to carve a little tunnel through the gel so that it can continue moving along the nerve to regenerate. And that bottom panel is an actual image of real living neurons. This was a laboratory experiment that we did. So this is a cluster of neurons growing within one of our protein engineered gels, and they're sprouting out these projections through the gel, and they're able to grow uninhibited and able to remodel this gel as they go along.
Okay. Now let me ask you this. How soon can we expect to see some products coming out of this?
Yeah, so for medical materials that would actually be placed in people, there is a long safety and regulatory process that's monitored by the FDA. But we are already beginning preclinical studies. Those are done in animal models of diseases and injuries. And that's the first step towards getting these things eventually into patients. So I would say if things go well in our animal studies, it's conceivable that in 5 years we could be to first in human trials.
And what's the hardest thing here? Is it engineering the proteins to your specifications, or figuring out what the specifications need to be to accomplish the objective?
I think both are really big challenges right now in the field. California is a real leader in regenerative medicine. We have the California Institute for Regenerative Medicine, and we're really beginning to learn a lot about these stem cells and how they can regenerate tissue, but we still don't really know what material cues or signals to give the cells to get them to unleash their full potential. So we're still trying to figure out what are the right properties to design into our material to coax the cells to do all of their regenerative function.
So you're doing a lot of basic physiology research, trying to find out how the body really works so you can figure out how to fix it.
It is very much an interdisciplinary set of experiments that we have to do. So we work very closely with colleagues at the medical school who are, for example, developmental biologists and disease physiologists, and they understand the biology and then they explain it to us, and then we try to incorporate that into our new material designs.